Welcome back to the deep dive. Today we are opening a file that is, well, it's honestly a little terrifying.
Yeah, it really is.
But it is also incredibly fascinating from a biological standpoint because we're looking at what happens when the human body's defenses just, go completely offline. We are diving into opportunistic mycoses.
Right, and it's a topic that really sits right at the intersection of microbiology and immunology. You really can't understand the fungus if you don't understand the specific flaw
Yeah,
in the host's defense.
exactly. And to guide us through this fungal jungle, we've got some really heavy source material today. Primarily, we are dissecting Murray's Medical Microbiology, Chapter 65.
The definitive text.
It really is our Bible for this session. But we've also pulled some supplementary clinical pearls from Jochalis to flesh out the diagnostic side of things.
It's a dense chapter. I mean, there's a lot of ground to cover.
It is. So let's set the stage. I want to start with this concept Murray introduces, which is essentially a host-pathogen chess match.
It's the perfect analogy for this.
Because normally, in a healthy person, the immune system is playing a grandmaster level game. It has the board completely locked down.
Right. In a healthy individual, you, me, hopefully the people listening, your innate immunity is doing all the heavy lifting. Your neutrophils, your macrophages, they are keeping the board clear.
Which is crazy to think about because we are breathing in fungal spores right now.
Literally, as we speak, they're landing on our skin. Candida is living in your gut at this very moment.
It's just hanging out.
Exactly.
Yeah.
But the immune system keeps them as pawns. They can't move. They can't advance.
But then opportunistic mycoses represent the moment the host loses their queen.
That is the exact moment of breakdown. When you lose that immune competence, whether it's through HIV destroying your T cells or, chemotherapy wiping out your neutrophils.
Or even something like uncontrolled diabetes, right?
Yes, even altering your blood chemistry creates a power vacuum.
Mm-hmm.
And suddenly, those pawns, those harmless environmental fungi or commensal yeasts, they realize the queen is gone and they storm the castle.
That's the distinction I want to nail down early for everyone. We aren't talking about the primary pathogens today.
No.
We aren't talking about the fungi that can hunt down a healthy lumberjack and take him out.
Right. Those are the endemic mycoses, things like histoplasmosis or blastomycosis. Those are true pathogens. They can infect absolutely anyone.
But today is different.
Today is strictly about the opportunists. These are the scavengers. They require a specific weakness to exploit. But the truly scary part is, once they find that weakness, they don't just nibble, they bite hard.
Okay, so we have a lot of ground to cover. Murray outlines what I'm calling the big four.
The non-negotiables.
Right, these are the major players you need to profile if you want to survive the exams, or honestly, the wards. Let's introduce them with some mental anchors right now just to keep them straight as we dive into all the details. First up is Candida.
Candida. Think of Candida as the insider. It's a commensal turned rogue. It's not flying in from the outside, it's already inside the house.
An insider, got it. Next we have Cryptococcus.
Cryptococcus is the armored invader. It's an encapsulated yeast. It wears this thick polysaccharide suit of armor that makes it incredibly difficult for the immune system to eat.
The armored invader. Okay, then we have the moulds. First, Aspergillus.
Aspergillus. I call this one the angioinvader. It's a mold that has a very specific predilection for eating through blood vessel walls. It's structural, it's sharp, and it's precise.
And finally, the one that honestly keeps surgeons up at night. Mucorales or Mucor.
Mucor. This is the blitzkrieg. It is rapid, destructive, messy, and aggressive. If Aspergillus is a sniper, Mucor is a bomb.
Okay, so insider, armored invader, angioinvader, and blitzkrieg. That really helps organize the chaos.
It gives you a framework.
And before we get into the biology of each, there's this overarching theme in Murray about the host factor. It seems like the type of infection actually tells you something about the patient's specific immune defect.
It does. It's almost diagnostic in reverse. If you see a specific fungus, you can almost guess exactly what's wrong with the patient's immune system.
Walk us through that. How does that work in practice?
Well, think about the layers of defense. If you see a mold like Aspergillus or Mucor invading tissue or invasive Candida in the blood, you usually think of a neutrophil defect. Neutropenia.
Because the neutrophils are the first responders.
Exactly. The police force that physically eats invaders is gone.
Okay, so molds equal neutrophil trouble.
Generally, yes. But if you see Cryptococcus or a mucosal candida like oral thrush, you should immediately think of a T-cell defect.
Because the T cells are more like the generals.
Right, that's the command center being offline. That's why we see those specific infections in advanced HIV and AIDS patients.
That is a crucial distinction to make. Neutrophils versus T cells. It's like knowing which branch of the military has been incapacitated based entirely on who is invading the border.
Precisely. It narrows your clinical focus immediately.
Okay, let's get into the weeds. Our first profile, the insider, Candidiasis.
Let's talk Candida.
So unlike the others, this is endogenous. It's part of the furniture.
It is part of the normal human flora. In the gastrointestinal tract, the female genital tract, it's on the skin. It's a neighbor.
But a neighbor that turns violent when the barriers break down.
Exactly.
Now, Murray lists a whole lineup of species, but we need to focus on the high-yield ones. Table 65.3 in the text is the key here. Obviously, we start with the heavyweight champion, Candida albicans.
Candida albicans, this is the classic. It accounts for 90 to 100% of mucosal isolates and a huge chunk of bloodstream infections.
So it's the most common by far.
By a landslide. If you hear Candida without a species name attached to it, you assume it's albicans until proven otherwise.
It's the default setting, but the landscape is shifting, right? It's not the only one we have to worry about anymore. We really need to talk about Candida glabrata.
Glabrata is the rising star, unfortunately. We see it a lot in older adults or people who've been on antifungals for a while.
And what's the big issue with it?
The key differentiator here, the thing you need to circle in your mental notes, is resistance.
Resistance to what exactly?
Azoles, drugs like fluconazole. Glabrata often has innate resistance, or it acquires it very, very easily.
So if you just throw the standard fluconazole at a glabrata infection, you aren't actually fixing it.
No, you might just be selecting for the strongest bugs and making the infection worse.
That's a critical clinical point. You can't just autopilot the treatment. Then there's Candida parapsilosis, which is a bit of a mouthful.
Parapsilosis. Think P for pediatric or P for plastic. This is the one we see a lot in the NICU, the neonatal intensive care unit. It loves catheters.
Why plastics, though?
It has this incredible ability to form biofilms on synthetic surfaces.
Let's unpack the biofilm concept for a second.
No.
Because it's not just the yeast sitting on the plastic, is it?
No, not at all. It
Wow.
secretes this complex slime matrix. It essentially builds a microscopic city on the tip of the catheter.
Like a fortress.
Exactly.
Right.
And that slime protects it from the immune system and, more importantly, from antifungal drugs. The drugs just can't penetrate the matrix.
It builds a bunker. So if you have a patient with a central line who suddenly spikes a fever, parapsilosis has to be on your radar.
Absolutely. You usually have to pull the line out entirely to cure it.
And finally, the scary new one, Candida auris.
The superbug. Auris is a major public health concern right now. It is emerging rapidly. It is multi-drug resistant, sometimes resistant to all three major classes of antifungals, and unlike other Candida species, it is highly transmissible in hospitals.
Wait, transmissible? I thought we just said Candida was endogenous.
And that is exactly why Auris breaks the rules. Albicans comes from your own gut. Auris sticks to hospital bed rails, blood pressure cuffs, the floors.
So it jumps from patient to patient.
It does. It causes massive outbreaks in ICUs. It's a complete nightmare for infection control teams because it is so incredibly hard to kill on environmental surfaces.
It's basically the hospital-acquired nightmare fungus. Okay, so we have the lineup. Albicans, glabrata, parapsilosis, and auris. Now let's talk identification.
Because on an exam or in the lab, you have to be able to tell them apart.
Right, and Candida is a bit of a shapeshifter, isn't it?
It is polymorphic, and this is actually a key virulence factor for Candida albicans. It's not just changing shape for fun.
What are the shapes?
Well, it can exist as a yeast, which are little round, distinct cells. These are great for dissemination, for floating through the blood and spreading.
Okay, the travel form.
Exactly. But it can also form pseudohyphae, which look like chains of sausage, pinched or constricted at the joints. And importantly, it can form true hyphae.
Which look different how?
True hyphae have long, parallel walls with no constrictions at the septations.
And the ability to switch between these forms is part of the attack.
It is. The yeast form travels, but the hyphal form invades. It literally uses the hyphae to punch through tissue barriers. It acts like a microscopic battering ram to get deep into the organs.
So how do we spot albicans specifically in the lab? Murray spends a lot of time on the germ tube test. This seems like a staple of every microbiology exam ever written.
It is the classic rapid test. What you do is take the yeast, put it in serum at 37 degrees Celsius body temperature for about 90 minutes.
And then what happens?
If it starts to grow a little tail, a germ tube, that is Candida albicans.
Just a tail.
It's actually the very beginning of a true hypha. It looks like a little sprout coming off the round yeast cell. Candida albicans and rarely a close cousin called Pathogenesis are the only ones that do this.
So everything else just stays as budding yeast.
Exactly. It's a quick yes or no for albicans. And it's incredibly cheap and fast.
And there's another specific test for albicans involving spores.
Yes, phlegmatospores. If you intentionally starve Candida albicans, say you put it on a nutrient-poor media like cornmeal agar, it forms these thick-walled survival spores called chlamydospores.
It's basically hunkering down for the winter.
Right, it's a stress response. So germ tubes and clamydospores equal albicans.
Now, what about glabrata? You mentioned it's the resistant one, but does it look different under the microscope?
Yes, and this is a great mental anchor. Candida glabrata. Think glabrous. Glabrous literally means smooth or bald.
Smooth.
Glabrata only forms yeast. It is simple. There are no pseudohyphae, no germ tubes, no true hyphae. Just smooth yeast.
Glabrata is glabrous. That is sticky. I really like that. So if you see a sample that is strictly yeast, but it's definitely Candida, and the patient isn't getting better on fluconazole,
you should be screaming glabrata and changing the antifungal.
got it. And briefly, chromagar. What is that?
It's a specialized agar plate that turns different colors based on the fungal species growing on it. Albicans turns green. Tropicalis turns blue. It's a very quick visual way to differentiate mixed infections in a single sample.
Green albicans.
Right.
Okay, let's move to the clinical side of things. What does candidiasis actually look like in a patient? We have the mucosal form, which everyone knows is thrush.
Right. This is the classic cottage cheese lesion. White plaques on the tongue, the palate, or down the esophagus. And the key clinical feature here, the differentiation point, is that it is pseudomembranous.
Meaning you can scrape it off?
Yes, you can physically scrape it off with a tongue depressor.
As opposed to what? What else looks like that?
As opposed to something like oral hairy leukoplakia, which is caused by the Epstein-Barr virus in HIV patients. Leukoplakia is integrated into the tissue. It does not scrape off.
So if you scrape thrush.
It comes off and leaves a raw, red, usually bleeding base underneath.
And the mechanism here is usually T-cell related, as we established.
Yes. Mucosal disease is almost always a T-cell deficiency. That's why we see it so frequently in HIV and AIDS patients, or even in babies whose immune systems are just immature.
But then there's the systemic form, candidemia.
This is the killer. This is when the organism gets past the mucosa and into the blood. The mortality approaches 50%. This isn't just an annoyance. This is a severe medical emergency.
And the mechanism shifts here from T cells to neutrophils.
Yes. Systemic disease is usually a neutrophil deficiency or a massive plethysmal breach. You've got a central line in where you've had major GI surgery and the gut flora just spilled into the peritoneum.
Once it's in the blood, where does it go?
What? Dissemination means it can go anywhere. It can go to the heart valves and cause endocarditis. It can go to the liver and spleen. But I want to highlight one specific location that Murray really emphasizes, and that is the eye.
The eye.
Endophthalmitis. Candida loves the eye. It seeds the retina and causes these distinct white cotton ball lesions.
That sounds dangerous.
It is. If you have a patient with candidemia, you must call ophthalmology for a dilated eye exam, even if the patient have absolutely no visual symptoms yet.
Because if you miss it.
If you miss it and the fungus scars the macula, they go permanently blind.
Wow. That is a massive high-yield clinical pearl right there. Check the eyes in candidemia. It's something you might easily overlook if you're just focused on stabilizing their blood pressure in the ICU.
Absolutely. And it changes how long you treat them, too. If it's in the eye, you treat for weeks longer just to ensure the drug penetrates the blood-retina barrier.
Before we leave Candida entirely, Jarvet's has a little tip on staining we should mention.
Yes. Candida doesn't actually stain very well with standard H&E, hematoxylin, and eosin. It can look kind of faint or ghost-like on the slide.
So what do you use instead?
If you really want to see it clearly in a tissue biopsy, you use a silver stain, like GMS or a PAS stain. That makes the fungus pop brightly, either bright magenta or stark black, against the background tissue.
Okay, that wraps up the insider. Now, let's fortify ourselves and look at the next one on the list. The armored invader. Cryptococcus.
Cryptococcus. This is a truly fascinating organism. It's a yeast, but it acts very differently from Candida.
It has a taxonomy split we need to understand first.
Yep.
Cryptococcus neoformans versus Cryptococcus gattii.
Right. Neoformans is the classic opportunist. It is found worldwide. And the association you absolutely have to know, it's the classic exam question, is pigeon droppings.
Why pigeon droppings of all things?
It's the nitrogen. The guano is incredibly rich in nitrogen, which the yeast just loves. Now, the birds themselves aren't actually infected. Their core body temperature is too high. It's around 42 degrees Celsius. And the fungus just can't survive that kind of heat. But once they drop the guano into the environment, it becomes a massive breeding ground in the soil.
So you really don't want to go sweeping up old barns or attics without a proper mask.
Exactly. The spores aerosolize very easily.
And what about C. gaudy?
Gaudy is the quote-unquote healthy pathogen, relatively speaking. It's traditionally associated with eucalyptus trees in the tropics, but recently we've seen major outbreaks in the Pacific Northwest, Washington, Oregon, Vancouver, associated with Douglas firs.
And it infects healthy people.
That's the scary thing about Gaudy. It can infect fully immunocompetent hosts. You don't necessarily need an immune defect to get taken down by it.
Interesting. So neoformans needs a weak host. Gaudy is a bit tougher. Now let's talk about why we call this one the armored invader.
The capsule. This is the defining structural feature of Cryptococcus. It is an encapsulated yeast. It builds this massive polysaccharide shell around itself made of a substance called GXM.
Glucuronoxylomanin. And this isn't just for, structural support.
No, it's a literal force field.
Yeah.
It is highly anti-phagocytic. Imagine a macrophage trying to eat a normal bacteria. It grabs it, engulfs it, and digests it. Now imagine that same macrophage trying to grab a wet, slimy bar of soap that is 5 times larger than its own hand.
It just slips away.
The capsule makes the yeast incredibly slippery and physically huge.
Yeah.
The immune system just can't get a grip on it.
Which leads perfectly to the classic diagnostic image, the India ink stain.
Yes, India ink is a negative stain. The ink itself is black and full of tiny carbon particles. The fungal capsule is so thick and so gelatinous that the carbon particles simply can't penetrate it.
So under the microscope,
You see this dark black background, the actual yeast cell sitting in the middle, and this bright, clear, glowing halo around the yeast. That halo is the physical space the capsule takes up, pushing the ink away.
but the sensitivity is pretty low, right?
Yeah, it misses about 50% of cases. So a negative India ink does not rule out Cryptococcus. But if you do see it, it's highly specific. It's a nice-to-know historical test, but not the definitive diagnostic for modern medicine.
There's another virulence factor here involving color. Melanin.
Cryptococcus actually produces melanin. It uses an enzyme called lacase to do it. If you grow the yeast on Niger seed agar in the lab, the colonies physically turn dark, brown, or black.
Melanin, like the pigment in human skin.
Very similar concept. In humans, melanin protects our skin cells against UV radiation damage. In the fungus, melanin protects it against oxidative stress from the immune system.
How does that work?
When a macrophage actually manages to corner the fungus, it tries to blast it with an oxidative burst, basically pouring bleach on it. The melanin physically absorbs those free radicals and neutralizes the damage. It is chemical armor layered directly on top of physical armor.
It really is built like a tank. Now, let's trace its journey through the body. How does it actually get in?
Inhalation. You breathe in the desiccated yeast cells or the basidiospores from that pigeon-dropping enriched soil we talked about. It goes straight down into the lungs.
And often in the lungs, it's just handled quietly.
In a healthy person, yes. Your alveolar macrophages and P cells wall it off. You might get a tiny asymptomatic granuloma, but in a T-cell compromised person like someone with AIDS, who has a very low CD4 count, it escapes the lungs.
It gets into the blood.
Gets into the blood, and then it exhibits this very specific, terrifying behavior called neurotropism. It loves the brain.
Why the brain? Why doesn't it go to the liver or the kidneys?
2 main reasons. One, the cerebrospinal fluid, the CSF, is a perfect growth medium for this specific yeast. It lacks complement proteins, and it lacks antibodies, so immune surveillance in the brain is incredibly low. It's a safe house.
And the second reason?
The organism has a specific affinity for the central nervous system, which is possibly related to that lacase enzyme we just mentioned. The brain is full of catecholamines, like dopamine. The fungus can actually hijack human dopamine and use it as a substrate to synthesize its melanin armor.
That is wild. It literally uses your own brain chemistry to build its defenses.
It is terrifyingly elegant biology.
So the clinical presentation we are looking out for is meningitis.
Chronic meningitis. It is not the lightning-fast bacterial meningitis that kills a patient in 24 hours. It's a slow burn. The patient have a headache, a low-grade fever, maybe some subtle personality changes, neck stiffness that builds over weeks.
But the really big complication is the ICP, the intracranial pressure.
Yes. Murray mentions a specific case study where the opening pressure on the lumbar puncture was over 400 millimeters of water.
Which is incredibly high. Normal is what, under 200?
Usually under 200. The mechanism here is purely physical. Because the capsule is so gummy, it constantly sheds that polysaccharide GXM into the CSF. It essentially clogs the physical drainage system of the brain, the arachnoid villi.
Like clogging a sink drain.
Exactly like clogging a drain with tapioca pearls. The spinal fluid keeps being produced, but it can't get out, so the pressure just builds and builds inside the skull. That pressure is what causes blindness, coma, and eventually death.
You also mentioned sentinel lesions on the skin.
Yes, this is an important clinical clue. Sometimes the very first sign isn't the headache, it's a skin lesion. It often looks exactly like molluscum contagiosum, a little raised bump with a central dimple and umbilication.
So it mimics a viral skin infection.
It does. If you see a lesion that looks like molluscum in an AIDS patient, you have to biopsy it. It might not be a virus, it might be Cryptococcus signaling that it is already disseminated through the blood.
Let's talk about the diagnosis hierarchy. How do we nail this down quickly?
The absolute gold standard today is antigen detection. We look for that capsular polysaccharide, the GXM, directly in the blood or the CSF using latex agglutination or an EIA. It is highly sensitive and highly specific.
Is it better than just culturing the fluid?
It's much faster than culture. Culture is definitive, of course, and you'll see these highly mucoid, snot-like colonies on the plate because of the thick capsules. But culture takes days to grow. Antigen testing takes minutes to hours. In a high-pressure situation, literally, you want the antigen test.
Okay, that's the armored invader. We've done the insider, we've done the armored invader. Now we pivot and we turn to the molds.
The filamentous fungi.
Section 3, the angioinvader, Aspergillosis. First clarification for the listeners, Aspergillus is a mold, not a yeast.
Correct. It is strictly filamentous.
Oh.
It exists as hyphae, and it is incredibly ubiquitous. You and I are breathing Aspergillus spores right now. They are in the air. They grow in shower heads. They are aerosolized at construction sites. You simply cannot avoid Aspergillus exposure.
So the exposure is constant. The major species we care about is Aspergillus fumigatus.
Fumigatus causes the vast majority of human infections.
Yeah.
But we also see A. flavus, A. niger, which is the black powdery mold you sometimes see on damp onions, and importantly, A. teresco.
Let's focus on morphology because this is the single best answer question waiting to happen on any board exam. How do we recognize Aspergillus under the microscope?
You look for the acute angle. Aspergillus has septate hyphae, meaning it has cross walls built into the tubes, almost like the segments of a bamboo stalk.
Yeah.
It's very uniform in its width.
And the branching.
It branches dichotomously, meaning one branch splits cleanly into two, and it does this at acute 45-degree angles.
The Z shape.
Exactly. Visualize the branches forming a strict V for victory, or A for Aspergillus in acute. It's a very rigid, structural, predictable branching pattern.
And what about the conidial head, that dandelion-looking thing?
You usually only see the fruiting head in a lab culture or in a patient if the fungus is growing inside an open cavity where there's actual air exposure. You rarely ever see the dandelion head in deep tissue invasion. In a deep tissue biopsy, you just see those V-shaped hyphae pushing through the cells.
Now, Aspergillus causes a wide spectrum of disease, entirely depending on the host. This is a perfect example of the host factor we talked about in the intro. It's the same bug, but 3 completely different clinical stories.
Exactly. With Aspergillus, it is all about the terrain, not just the pathogen itself.
Scenario 1, the hyperimmune host, the allergic patient.
This is ABPA, allergic bronchopulmonary aspergillosis. This typically happens in patients who already have asthma or cystic fibrosis.
So the fungus is invading them?
No, the fungus isn't invading the tissue at all. It's just sitting there, trapped in the thick mucus in the airways. But the host's immune system absolutely freaks out. It mounts a massive inappropriate hypersensitivity response. You get high IgE levels, severe eosinophilia.
It's basically a severe allergic reaction.
Right, like the worst hay fever imaginable, but happening deep inside the bronchi. It causes severe bronchospasm and these dense mucus plugs that block breathing.
So because it's an allergy, not an invasion, the treatment is different.
You primarily treat with systemic steroids to calm massive inflammation. You might add antifungals just to reduce the overall fungal burden triggering the allergy.
Okay, scenario 2, the structural defect. The patient with an old TB cavity.
This is the Aspergillus, also known as the fungus ball.
That sounds lovely.
It's exactly what it sounds like. The fungus spores get inhaled, and they find a pre-existing hole in the lung tissue, usually a cavity left over from old tuberculosis or maybe severe sarcoidosis. The fungus just sets up camp inside that empty space. It grows and tangles into a literal physical ball of hyphae.
Is it invading the surrounding lung tissue?
No, it's just colonizing the empty space.
Yeah.
It physically rolls around inside the cavity. But because it is moving, it can slowly erode the blood vessels in the cavity wall just by friction or by secreting local enzymes.
Which causes what?
Hemoptysis. The patient starts coughing up blood, sometimes massive, life-threatening amounts of blood.
And radiologically, you can actually see this ball.
Yes, and it is gravity-dependent. If you roll the patient over onto their stomach while they're in the CT scanner, the fungus ball will physically drop and roll to the new bottom of the cavity. It is completely mobile.
That's wild. And there's a specific term for the X-ray finding.
The air crescent sign. You see a solid, opaque ball, and right above it, a thin sliver or crescent of dark air.
Okay, scenario 3. The immunocompromised host. The profoundly neutropenic patient.
This is invasive aspergillosis. This is the absolute killer. This happens when the neutrophils, the only police force capable of stopping the mold, are completely gone, usually from chemotherapy or bone marrow transplants.
And this is where that nickname, the angioinvader, finally comes into play.
Exactly. Without neutrophils to stop its advance, the hyphae don't just grow passively in the air spaces. They actively punch their way through the lung tissue and physically invade the walls of the pulmonary blood vessels. They grow straight into the lumen of the vessel.
What happens when mold grows inside a blood vessel?
It instantly clogs the vessel, triggering massive thrombosis or blood clots. That clot completely cuts off the blood supply to the lung tissue downstream. So you get pulmonary infarction tissue death and rapid necrosis. The lung tissue literally starves of oxygen and dies because the fungus choked off the blood supply.
That is incredibly grim. And there's a specific radiologic sign on the CT scan for this invasion, too.
The halo sign. On a chest CT, you will see a dense central nodule, which is the actual fungal infection surrounded by a fuzzy ground glass haze.
What is the haze?
That haze is hemorrhage. It is literal bleeding into the surrounding lung tissue because the fungus is destroying the integrity of the invaded blood vessels.
Let's talk about a couple of testing gotchas for the exam here. You mentioned Aspergillus terreus earlier.
Yes, A. terris is the rebel of the family. It is intrinsically resistant to amphotericin B. For decades, Ampho B was the absolute gold standard, the gorilla antifungal we use for everything severe. If you have an A. terris infection, Ampho simply will not work.
But how do you know it's terris without waiting days for a culture sensitivity report?
In a tissue biopsy, terris produces these very special spores called Candida right there in the tissue.
Wait, I thought you just said we don't see spores in tissue with Aspergillus.
We usually don't.
Yes.
That's the rule. But terris is the specific exception that breaks the rule. If you look at a biopsy and you see the V-shaped hyphae, but you also see spores scattered around in the tissue, you have to think terris, and you must not give amphotericin B.
Another key diagnostic test, galactomannan.
Galactomannan is a structural carbohydrate component of the Aspergillus cell wall. We can run an antigen test for it in the patient's blood or in the bronchoalveolar lavage fluid. It is an absolutely fantastic marker for early invasive disease in neutropenic patients.
Why do we need the antigen test? Why not just draw blood and culture the fungus?
Because blood cultures for Aspergillus are notoriously negative. The fungus is physically stuck, anchored inside the blood vessel wall or deep in the necrotic tissue. It very rarely floats freely in the bloodstream the way a yeast like Candida does. So you can't just draw blood and grow it. You have to look for the pieces of its wall floating around. You need the galactomannan test.
Okay, that wraps up the angioinvader. Now we move to the final member of the Big Four, the one that truly scares the surgical teams. The blitzkrieg, mucormycosis.
The order Mucorales. This primarily includes the genera Rhizopus, Mucor, and Leishmania.
These are the common bread molds, right?
Yes, highly environmental. They live in soil, on decaying fruit. If you leave a loaf of bread out on the counter and it gets that incredibly fuzzy, fast-growing gray mold, that is very often a Mucorales species.
Morphology is the absolute key differentiator here from Aspergillus. You have to be able to tell them apart immediately on a pathology slide.
You do, because the treatments are opposite. Mucor is broad and aseptate.
Aseptate meaning it lacks those cross walls we talked about.
Right, it is coenacidic. The hyphae are just long, hollow, open tubes with nuclei flowing freely inside. Because there are no internal walls to provide structural support, the hyphae look very floppy under the microscope. They create a very distinct ribbon-like appearance. The hyphae physically fold and twist over on themselves because they lack rigidity.
Kind of overall width.
Massive, usually 10 to 15 microns wide. It is much, much wider than Aspergillus hyphae.
And what about the branching pattern?
It is irregular, non-progressive, and typically branches at wide 90-degree right angles.
Okay, let's pause and visualize this side by side. Aspergillus is narrow, septate, with neat 45-degree acute angles, V for victory. Mucor is broad, Aseptate, floppy, and ribbon-like, with messy 90-degree right angles.
That is the perfect mental differentiation. If you mix those two up on a slide, you will prescribe the wrong drug and the patient will almost certainly die.
Now let's talk risk factors. The why. Why does this harmless bread mold suddenly decide to become a flesh-eating nightmare?
There are two main risk groups that Murray highlights. First, and most famous, is DKA, diabetic ketoacidosis.
Why DKA specifically? What is it about that state?
This is a beautiful but deadly bit of biochemistry. Rhizopus has a specific enzyme called ketone reductase. It allows the fungus to survive and actually thrive in a highly acidic, high glucose, high ketone environment.
Which is exactly what DKA is.
Exactly. In a normal, healthy body, the blood pH is neutral, and the serum proteins tightly bind up all the free iron, which prevents the fungus from growing. But in DKA, the patient's blood becomes very acidic. That acidosis literally causes iron to unbind from transport proteins like transferrin.
So suddenly there's free iron floating around, plus massive amounts of blood sugar.
Yes. The iron acts as a growth factor, and the sugar feeds the fungus. It's an all-you-can-eat buffet for Rhizopus.
Okay.
It is opportunistic in the most literal biochemical sense.
So it feeds on the metabolic crisis. What is the second major risk factor?
Iron overload. As we just established, this fungus is ferrophilic and absolutely loves iron, needs a lot of iron to grow rapidly.
So patients with a disease like hemochromatosis are at risk.
Yes. Or, more classically on exams, patients who are on a specific drug called deferoxamine.
What is that?
Deferoxamine is an iron chelator. It's a drug given to patients with iron overload to bind up the excess iron and help them excrete it. But biologically, Rhizopus is incredibly smart. It recognizes deferoxamine and uses it as a siderophore.
Meaning what?
It basically steals the iron right off the drug molecule and pulls it inside its own fungal cells to fuel its growth.
So the drug that is literally meant to safely lower the patient's iron actually gathers it up and delivers it to the fungus on a silver platter.
Exactly. It's a tragic irony. We use different, safer iron chelators now, primarily because of this specific risk, but it remains a classic board question.
Let's get to the clinical presentation. Rhinocerebral mucormycosis. This is the stuff of actual nightmares.
It really is. This is the classic presentation in an uncontrolled diabetic patient. They come into the emergency room with severe facial sinus pain, maybe some bloody or dark nasal discharge, and swelling around one eye. Initially, it just looks like a really bad bacterial sinusitis.
But then you look inside the nose.
And you see the black eschar, a black, necrotic, dead patch of tissue on the hard palate or on the nasal turbinates.
What exactly is that black stuff?
That is dead human tissue. Just like Aspergillus, Mucor is highly angioinvasive. It rapidly invades the blood vessels of the face and infarcts the tissue. But it does it at blitzkrieg speed. The tissue dies, loses blood supply, and turns completely black.
And it doesn't just stop at the nose or the palate, does it?
No, it burns through tissue indiscriminately. It goes right up through the cribriform plate, the thin bone at the roof of the nasal cavity, and grows straight into the frontal lobe of the brain. It causes cavernous sinus thrombosis, massive brain abscesses. It is rapidly, almost inevitably fatal, if not caught early.
So treatment. You said earlier this is scary for surgeons.
It is a surgical emergency. You absolutely cannot cure mucormycosis with antifungal drugs alone.
Why not?
Because the tissue is already dead, and the blood vessels leading to that tissue are entirely clotted off by the fungus. So even if you pump the patient full of IV antifungals, the drug physically cannot reach the leading edge of the infection because the blood supply is blocked. You have to cut the dead tissue out.
Debridement.
Extremely aggressive surgical debridement. We're talking about removing the hard palate, souping out the sinuses, sometimes removing the entire orbit in the eye or taking off parts of the face. It is horribly disfiguring surgery, but it is the only way to save their life. If you do not cut out all the infected tissue, the fungus reaches the deep brain and the game is over.
And what drug do you give alongside the surgery?
Amphotericin B. High dose, usually a liposomal formulation to protect the patient's kidneys.
Does voriconazole work here? We use that for Aspergillus all the time.
Absolutely not. Voriconazole has zero activity against Mucorales. None. This is a very classic exam trap and a fatal real-life medical error. If you mistake Mucor for Aspergillus and give voriconazole, the patient will die. In fact, we actually sometimes see Mucor breakthrough infections in leukemia patients who are actively taking voriconazole prophylaxis. The drug effectively kills all the other competing fungi, leaving the biological field wide open for Gaiputa to take over.
Wow. So correct identification under the microscope is literally life or death. Aspergillus gets voriconazole, Mucor gets amphotericin B. Do not mix those up.
Precisely. It is the most critical distinction in medical mycology.
Okay, those are the big four. Let's do a very quick flyover of Section 5 for Murray, the other opportunist. Just some high-yield associations we need to mention. First, fusarium.
Fusarium. This is another environmental mold. Morphologically in tissue, it actually looks a lot like Aspergillus. It has septate branching hyphae.
So how do we tell them apart if they look the same in tissue?
You have to culture it. If you grow fusarium on a plate, it produces these very distinct, large, banana-shaped or canoe-shaped macroconidia.
Banana-shaped. And what are the clinical associations for fusarium?
Think about contact lens wearers. It is a major cause of fungal keratitis. If a patient who wears contacts comes in with a really nasty treatment-resistant corneal infection, you have to think fusarium. It can also cause disseminated severe disease in major burn victims.
And the drug resistance profile.
Highly, highly resistant. It is notoriously difficult to treat effectively.
And finally, pneumocystis jirovecii. We usually cover this in the dedicated HIV or pulmonary chapters, but it is technically an opportunist.
It is a major opportunist. It's strictly an AIDS-defining illness, typically showing up when the CD4 count drops below 200. It's technically classified as a fungus based on its DNA, but it acts very weirdly.
How so?
It completely lacks ergosterol in its cell membrane.
Which is the target for most antifungal drugs, right?
Exactly. So amphotericin B, which binds ergosterol, or azoles, which block ergosterol synthesis, simply do not work against pneumocystis.
So how do you treat a fungal infection if antifungals don't work?
You actually treat it with antibacterial drugs, specifically TMP-SMX, or Bactrim. It targets the folate synthesis pathway instead.
Okay. We have unpacked a tremendous amount of biology today. Let's try to bring it all together in Section 6 with a clinical pattern recognition summary. I call this the host pathogen matrix. I will give you the rapid clinical picture, and you tell me the fungus.
Let's do it.
Scenario 1. A patient has acute leukemia, severe neutropenia after chemo, a high fever, and a CT scan shows a lung nodule with a clear halo sign.
Think Aspergillus, or possibly Mucor. But Aspergillus is statistically much more likely with the classic halo sign. The neutrophils are gone, so the mold takes over.
Scenario 2. An AIDS patient with a CD4 count of 50 presents with a severe headache, confusion, and neck stiffness that has been building for 3 weeks.
Cryptococcus. A severe T-cell defect plus slow-building CNS symptoms equals cryptococcal meningitis until proven otherwise.
Scenario 3. An AIDS patient complains of pain when swallowing, and you see thick white plaques all over their tongue that easily scrape off.
Candida. That is classic mucosal candidemia, or thrush.
Scenario 4. A diabetic patient arrives in severe ketoacidosis, complaining of facial pain, and you notice a black necrotic spot on the roof of their mouth.
Mucormycosis. That black escher is the absolute giveaway. It is a surgical emergency.
Scenario 5. A patient who has had a central venous line in the ICU for 2 weeks suddenly spikes a fever. The blood cultures come back positive for yeast.
Candida. It is probably albicans, but given the central line, it could very well be parapsilosis forming a biofilm, or glabrata. Central lines are absolute superhighways for Candida.
This kind of pattern recognition is exactly what saves lives on the wards. You see the pattern, you immediately order the right test, and start the right empiric therapy.
Exactly. You cannot wait for the final culture to come back 3 days later to start thinking about what it might be. The patient will be dead. You have to suspect it right now based on the host.
All right, listeners, get your mental pencils ready. It is time for Section 7, our learning reinforcement phase. We are going to do a rapid single best answer quiz.
I love this part. This is where we actively solidify the models we just built.
I will read the clinical vignette. I'll pause for a couple of seconds to let you think about it. Then our expert will reveal the answer and, most importantly, explain the why. Here we go. Question 1. A 45-year-old male who is HIV positive presents with a severe headache and confusion. A lumbar puncture is performed. The CSF fluid shows encapsulated yeast. Which stain is the most specific for visualizing that capsule?
The answer is the India ink stain, or musicarmin.
And why?
Because the massive polysaccharide capsule physically excludes the black ink particles, which creates a highly visible glowing halo around the yeast cells under the microscope. This is the classic morphological feature of Cryptococcus.
Question 2. A blood culture drawn from a patient with a central line grows yeast. The microbiology lab incubates the yeast in serum at 37 degrees Celsius for 90 minutes, and the yeast produces germ tubes. What is the specific species?
The answer is Candida albicans.
Why?
The germ tube test is the rapid lab differentiator. Candida albicans and very rarely doubliniensis are the only ones that form germ tubes. Other clinically relevant species like glabrata or tropicalis simply do not.
Question 3. A patient with leukemia has a persistent fever and lung infiltrates. A biopsy of the lung tissue shows septate hyphae that branch at regular 45-degree angles. Name the organism.
The answer is Aspergillus species.
Why?
The morphology is the absolute key here. Septate hyphae with cross walls plus acute 45-degree branching equals Aspergillus. Remember, if it were 90-degree right angles and aseptate, it would be mucor.
Question 4. A patient in diabetic ketoacidosis presents with severe facial pain and a black necrotic patch on their hard palate. A biopsy of the tissue shows broad aseptate hyphae. Name the disease.
The answer is mucormycosis, caused by rhizopus or similar molds.
Why?
The combination of the DKA risk factor, meaning high acid and high sugar freeing up iron, and the black Escher is the classic clinical presentation. The morphology of broad aseptate ribbon-like hyphae confirms it. Remember, this requires immediate surgical debridement.
Question 5. An elderly patient grows a yeast from a blood culture. The lab notes that the organism does not form pseudohyphae, it only forms yeast cells, and susceptibility testing shows it is highly resistant to fluconazole. What is the species?
The answer is Candida glabrata.
Wow.
Recall the mental anchor we set. Glabrata is glabrous, meaning smooth. It only forms simple yeast cells, never pseudohyphae or true hyphae. And the strong, innate azole resistance profile is the clinical kicker that should immediately point you to glabrata.
Question 6, the last one. A severely neutropenic patient has a rapidly invasive fungal lung infection. The pathologist notes that the organism produces Alluria candida directly in the tissue biopsy, and the infection is found to be completely resistant to amphotericin B. Name the organism.
The answer is Aspergillus terris.
Why?
This tests the specific exception rule. Terris is the only major Aspergillus species that is intrinsically resistant to amphotericin B, and the physical presence of spores, the Alluria candida, directly in the deep tissue is biologically unique to this specific species.
How did you do? Hopefully those mental anchors we talked about, the V-shape, the black Escher, the germ tube, really helped you nail those answers.
And if you missed any, just go right back to Murray chapter 65. Look closely at the images. Medical mycology is a highly visual sport. You really need to see the angles and the shapes with your own eyes to remember them.
We're getting near the end of our time. Expert, do you have a final provocative thought to leave us with today?
I do. We spent this whole time talking about these fungi strictly as opportunists, right? Scavengers that are just passively waiting for our immune systems to get weak. But the global landscape is changing. We are actively seeing the rise of emerging primary pathogens. Fungi are not static organisms. They are evolving rapidly.
Evolution happening in real time.
Exactly. Just look at Candida auris. It literally didn't exist in the medical literature a few decades ago. Now it's a massive global threat spreading through ICUs. Why did it suddenly appear? Some major evolutionary theories suggest climate change is the driver.
How does climate change create a superbug?
As the globe slowly warms, these environmental fungi are being forced to adapt to higher and higher ambient temperatures. Historically, the human body temperature of 37 degrees Celsius was a thermal barrier. It was too hot for most fungi to survive inside us.
So the thermal barrier that used to naturally protect us is eroding.
Potentially, yes. They are getting used to the heat. And when you combine that thermal adaptation with the massive, widespread use of agricultural fungicides, azole drugs sprayed constantly on commercial crops, we might be selecting for heat-tolerant, highly drug-resistant molds out in the environment long before they ever enter a human host. Are we accidentally engineering our own next plague? It's definitely something to think about.
That is a chilling but incredibly important thought. The chess match continues, but the other side is learning entirely new moves.
Always. You can never underestimate them.
For everyone listening, your call to action today is simple. Open up Murray chapter 65. Look at the full-color image of the germ tube. Look at the India ink stain showing that glowing halo. Look at the physical hyphal branching angles of Aspergillus versus Mucor. Do not just read the text, physically burn those images into your retina. They are guaranteed points on your exam, and they will absolutely help you diagnose a real patient in distress.
See it, know it, treat it fast.
Thanks for diving deep into the source material with us. We'll see you on the next one.
Stay safe out there.